Hello and welcome all of you to T10 by Bangalore IAS Academy. In this video, we are going to discuss a very important and a very sought after concept from science and technology, the CRISPR-Cas9 technology. Now, CRISPR-Cas9 is something that is constantly being confusing the candidates. How exactly is this technology? How deep I should study this? It's actually a Nobel Prize winning technique, but it's a very simple technique. If you listen to me for the next few minutes, I'm going to clear all the confusions with respect to this technique, CRISPR-Cas9. Now, why we have picked it up for T10? Because it was repeatedly being asked by UPSC. You can see CRISPR-Cas9 being asked. So, what exactly is this? CRISPR and Cas9. CRISPR-Cas9, first thing to understand, is a gene editing technology. So comes the question, what is a gene? Now to explain a gene, think of it like this. Why is your height this much and not 6 feet or 7 feet? Why is your color like this and not like someone sitting in US or Europe? Why is your nose like this? Why is your eyes like this? Why is your hair like this? Now why is the difference? What makes this difference in human beings? Or think of animals. What makes every animal unique in their own way? Something needs to be there. So that something is called the gene. Gene is that information that determines the way your functions of the body is. So in every cell of your body, you know that your body is made up of cells. Everywhere you have cells. In your hair, in your eyes, everywhere you have cells. In every cell, you have something called DNA. Within the DNA, you have a gene. Gene, in simple words, is genetic information. Based on this information only, it is determined how should be your color, how should be your everything. So you resemble your parents because your gene comes from your parents. Their gene comes from their parents. So gene determines everything. Gene is that information. Now, diseases can be caused by gene. Diseases can be cured by genes. So, editing the gene is a very key technology. And CRISPR-Cas9 is one tool that will help you to edit this gene. That is the relevance of it. Now, I hope you understood what is CRISPR-Cas9 and what is the basics of it, biotechnology. Now, how did we find it? Did human beings just invent a machine which can edit human gene? No. This is one technology that was present in bacteria. Bacteria has been using it for a long time. Now, why would bacteria use it? Listen to me carefully. Let's say a virus. You know virus, right? Virus doesn't have its own genetic information. Virus have information, but virus doesn't have life. Virus has to attach itself to a host cell for it to function. Okay? So COVID doesn't have its own life, but it has some genetic information. Only when it is attached to a human host, it will take action. So just like that, virus can infect a bacteria also. So what would bacteria do? Let's take a virus. Virus A. Let's name it like that. This virus is infecting a bacteria. Once this virus infects a bacteria, what will the bacteria do? So this virus also has genetic information. The bacteria will cut a small part of this genetic information and they will store it. So next time the same virus comes, this bacteria knows how to defend it. This is the whole idea of vaccine and immunity, right? You are infecting, so when you have a particular vaccine in your body, you are infecting your body with little bit of that disease so that your body can resist it better. So the bacteria have been using it for a long time. And how the bacteria did it? They did it by cutting the gene, cutting the DNA or RNA of that particular virus. This is the key. Bacteria knew how to cut the genetic information. So from there, we took that technique and we developed it into much higher scale. That is CRISPR-Cas9. So CRISPR is one technology that was there in bacteria, and we have taken this technology from them and we started using it for gene editing in general. That's how it is. Now, what is Cas9? So before going into Cas9, I'll just show you the expansion of CRISPR. CRISPR is not a very funky term. CRISPR has an expansion. Clustered, regularly, interspaced, short, palindromic repeats. That's the full form of CRISPR. I don't recommend you to remember this. Just keep in mind it has an expansion. Sometimes UPSC might give you this expansion only. So just be familiar with it. That's all I'm saying. Now let's come to Cas9. So to understand Cas9, you should understand the process of how gene editing is done. So let's take one example. Let's say in your body, there are billions of genes. There are that many DNAs. How can I cut a very specific DNA? You cannot even imagine it. It's very difficult. Your body is so huge and DNA is so small. How are you going to pinpoint on a very particular genetic piece? It's very difficult. That is where Cas9 comes into picture. Cas9, in simple words, is called a molecular scissor. So Cas9 is the scissor that is going to precisely cut the very specific area in your gene which needs to be it. Now, how will the Cas9 go there in the first place? This Cas9 needs to be guided by someone. That guide is the guide RNA or the gRNA. So CRISPR is the cutting technology, Cas9 is the molecular scissor, and Cas9 is taken to the DNA by the guide RNA. Cas9 will go there, guide RNA will take the Cas9, Cas9 will cut the material, and after cutting, it can do addition, it can do deletion, and it can do nothing also. You can just take it out. So this is the process. Just like the constitutional amendment, addition, deletion, repealing, Cas9 can cut it and it can do what is needed. But you can think, what if genetic information is randomly cut out from the body? What is going to happen to the body after this? Don't worry, the body has its own repairing mechanism. So the DNA's own repairing mechanism, the cell's own DNA repairing mechanism will come into play, and this will be alright. So that is also playing a function here. It's not a completely artificial process. Own repairing mechanism plays a big part. So in short, CRISPR-Cas9 is a gene editing technology. CRISPR is a technique that was found in the bacteria. From there we have scaled it, we have developed it. So to cut the specific part of the DNA, Cas9 is used, which is the molecular scissor. To guide the Cas9 to the particular DNA, the guide RNA is used. And once the cutting is done, you can add something there, you can delete something from there, or you can replace something was there. And the cell's own DNA repairing mechanism will come into play to cure it afterwards. So this is CRISPR-Cas9. Thank you. See you in the next video with some other topic.